Alright, this is the effects of the 5-second rule and the growth of bacteria on apples. My name is Morgan Reinhardt. My course is Basic Microbiology Lab, BI 150. My instructor is Lori Manzel. I'm in Lab Section B, and the date was March 29th. So my introduction purpose was to test the authenticity of the well-known 5-second rule on apples dropped on the floor. People often use the 5-second rule as a rule of thumb when it comes to food, disregarding the number of bacteria that can be picked up and the amount of time. Everyone has used the 5-second rule once or twice in their life, and everyone disregards that you can pick up really harmful bacteria on the ground and then ingest it later. People often tend to overlook that it doesn't matter the amount of time the food is on the ground for, but that how long or what kind of bacteria is on the ground. Our group posed the question, if we drop an apple slice on the ground, will it pick up, if any, bacteria compared to the slice that was not dropped on the ground? We hypothesized that the apple slice that was dropped on the ground would have more bacterial species growing on it than the slice that was not dropped on the ground. Our methods: the supplies needed were sterile water, 2 cotton swabs, 1 apple slice, a sterile knife to slice the apples, 2 nutrient augers, and gloves. The apples were sliced with a sterile knife. Next, we dipped the cotton swab into the sterile water and swabbed a side of the apple that had not been contaminated yet. Then we dropped the opposite side of the apple slice face down to the floor and left it on the floor for 5 seconds. Picking the apple up with a pair of gloves, we then used a new cotton swab, dipped it in sterile water, and swabbed the new side of the apple that had been face down to the floor for 5 seconds. Using the streaking technique, we streaked both apple sample swabs onto their designated auger plate. The plates were left to incubate for 24 to 48 hours, and our control was the apple slice that was not dropped on the ground. After leaving 1 apple untouched and dropping 1 onto the ground for 5 seconds, we left the individual augers to incubate. A trend that we saw after reviewing our experiment was that the apple that was dropped on the ground grew more organisms than the apple that was left alone. Each time this experiment was repeated, the apple that was on the ground grew a substantial amount of bacteria compared to the slice that was not dropped on the ground. This is a graph that shows bacterial growth on apple slices using the 5-second rule. This is the bacterial species on the apple slice that wasn't dropped on the ground. And this is the bacterial species on the apple slice that was dropped on the ground. You can see that there was a lot more bacterial growth on the one that was dropped on the ground, and this was true for all 3 experiments that were performed. Our hypothesis was supported. This is significant because everyone has heard or has followed the 5-second rule, which disregards the bacteria that is actually grown. Bacteria thrives in unclean environments, Jane 2019. Your floor can have a lot of harmful bacteria on it that you don't even know about. Your floor cleaners could not be working as efficiently than you think that they are. There can be a lot of bacteria on the ground anywhere that if you drop food on the ground, it could pick up. By ingesting food that has been on the floor, you have a chance of picking up harmful bacteria. You could pick up bacteria like E. coli, which is a gram-negative bacteria, Staphylococcus, which is a gram-positive bacteria, or many other harmful organisms. An error that occurred in our experiment was that one of the augers was left face down, causing moisture to affect the bacterial growth. This auger was not included in our experiment, however, because it wouldn't give us an accurate result. If I could do this experiment again, I would make it more specific. I would try maybe dropping food on the lunchroom floor or floors that are more commonly traveled, like hallways or staircases that people use a lot during the day, not just the laboratory floor that is seen by not as many students a day. You could try this experiment with different foods or in different areas, and I would hypothesize that the results would still be the same, because there's going to be more bacteria on the floor than bacteria that is just on a piece of food that hasn't been dropped on the ground or that hasn't been contaminated in any way. Okay. The next step for this would be to try and drop it on different floor locations and observe the bacterial growth differences between the different floor locations or different foods. In conclusion, we found that when food is dropped on the floor, it grows more bacteria than when the food is not dropped on the floor. This is what we hypothesized, and it was supported. Here's my literature cited. The primary source is the first. The books that I used are cited, and that's all. Thank you.